I'll be telling you a story about Anna and the cat. Anna is a little girl who likes to play with building blocks. She likes to build anything tall, bridges, tall building, tall castle, and she has a cat. The cat, on the other hand, likes to destroy whatever it is that she is building. So the cat will destroy her tall building into smaller pieces. So I want you to think about this Anna and the cat at the back of your mind whenever we're going over anabolic reaction and catabolic reaction. So there are two types of enzyme actions, anabolic and catabolic. What are they? Okay, we've talked about lock and key hypothesis, whereby the substrates that are complementary to the active site of the enzyme can fit perfectly and form this enzyme substrate complex. And during anabolic reaction, the enzyme slightly joins the substrates, combining the substrate and making one new product molecule. And once the product is being formed, the product will be released, and now you have free active site on the enzyme, and this enzyme can be reused to react with another new sets of substrates to form another anabolic reaction by forming this enzyme substrate complex. And this takes place over and over again until all of the substrates has been used up. One example of photosynthesis is, one example of anabolic reaction is photosynthesis. So even though these are the equation of photosynthesis that I'm sure you know by now, these are the raw materials that you need for photosynthesis, and these are the products of photosynthesis. So even though enzymes were not mentioned in this equation, actually enzymes are involved in the process of photosynthesis to speed up the process of photosynthesis. And again here, the role of the enzyme as anabolic reaction, whereby the enzyme will be joining these two substrates, forming this new bigger product molecule. Okay, what about catabolic reaction? Catabolic reaction is the opposite of the anabolic reaction. So again, the substrate that is complementary to the enzyme will fit perfectly into the active site because enzyme again is specific. So forming this enzyme substrate complex. For catabolic reaction, the enzyme here slightly bends the substrate. When it does that, the enzyme split the substrate, forming two new product molecules. And these products will be released by the enzyme, and again the enzyme now has a free active site which is available to join with a new set of substrates, forming enzyme substrate complex, and then splitting the substrate to form two new products. And this process goes on and on until all of the substrate has been used up. And because the enzymes can be reused over and over again, you don't actually need a lot of enzymes in a reaction. So one example is the breakdown of hydrogen peroxide that you have come across this plenty times before. Another example of catabolic reaction is digestion. You will come across this when you are learning about topic 5, animal digestion. So the example is the breakdown of protein into smaller molecules. The final product is amino acids, and the enzyme that you need is pepsin. The other type of substrate in digestions are starch, which is what we can find in the rice or in ambuyat, maltose that we can find in sweeties, and also fats. And these are the enzymes that is used to break these down into smaller molecules so that these can then be absorbed into your blood. Once it's absorbed into your blood, it can then be carried to the cells and can then be used up to make parts of the cells that the cells need to make. For example, in the cells, there are proteins. In the cells, there are fatty layer, or the cell membrane. So, going back to Anna and the cat, I would like you to think about anabolic reaction and catabolic reaction, just like the story of Anna and the cat.